Welcome, everyone. Thank you for joining us to discuss the recent developments on the UQ COVID-19 vaccine. My name is Jennifer Karlson. I'm the Pro-Vice-Chancellor of Advancement at UQ. Today, I'm joined by Professor Paul Young and Professor Trent Munro from UQ's COVID-19 vaccine project. On behalf of all of us, we'd like to begin by acknowledging the traditional owners and their custodianship of the lands on which we meet today. We pay our respects to both ancestors and descendants who continue cultural and spiritual connections to country. And of course, we recognize their valuable and ongoing contributions to Australian and global society. So, here we go. For the past 11 months, the UQ COVID-19 vaccine development team have worked around the clock in the bright spotlight of a world waiting for hope. Your encouragement and has been absolutely steadfast and a huge source of inspiration for this team, who have worked tirelessly to progress this vaccine. As you would have known by now, last week there was an unexpected finding in the phase 1 trial where participants generated a low-level antibody response to fragments of a protein called GP41, and that's used in the molecular clamp technology. That finding ultimately led CSL and Australian government's decision to not progress the UQ COVID-19 vaccine to phase 2/3 clinical trials. This is, of course, really disappointing, but while the UQ COVID-19 vaccine is unlikely to be rolled out to fight this global pandemic, today I hope we'll hear a little bit more about the learnings that have occurred and how their really incredible efforts might better prepare us for the future. So, Paul and Trent, we're so grateful you've taken the time to join us today to answer some of the questions from our UQ community. Welcome to you both. I might begin by saying on behalf of UQ and all of us who are joining today, I'd like to start by saying thank you for your efforts this year, and of course to you, our members of our alumni and donor community who have not only submitted questions today, thank you for sharing your heartfelt messages of support from really around the world. And I know you're getting a lot of those directly, Paul and Trent. So let's jump in, shall we? Let's see, we've got our first questions coming from Alan Say. Alan asks, what happened and what caused the team to abandon the vaccine work? Paul, could I ask you to talk a little bit more about that?
Yes, of course I can. Thanks, Jen. Maybe before I start on that question, I'd just like to also add my thanks to the UQ community and the broader community for that matter for their unwavering support over the period of time that we've been on this journey to develop the vaccine. It really has been extraordinary, and the support, it has been an inspiration for the group, on those long nights and on those long weekends. It's been 11 months of solid work now, and we've come a long way, and we really appreciate that very, very strong support. So where we'd reached with that development pathway was towards the end of the business end of a phase 1 clinical trial, when we unblind the data and get to see what the responses in our participants were to that vaccine. And it was extremely encouraging. We saw that the vaccine was safe and well tolerated, possibly better than some other vaccines that are out there at the moment, in that minor pain and tenderness around the injection site was pretty much all that our participants experienced. But more importantly, the immune response that was elicited at every dose that we tried was strong neutralising antibody response, which was able to kill the virus in cell culture in the lab. And it was at a level that was above what we saw in the antibody responses in recovered patients. So we were hitting the mark in terms of what we were trying to elicit. As I said, all of the doses we tried elicited similar responses, which means that the manufacturing of the vaccine could be spread further should it be deployed at the lower doses that we showed were strong and elicited that good response. The other really important finding was that we were seeing just as strong a response in our elderly cohort as we saw in our younger cohort. So, all of the milestones we set for ourselves were hit, if not exceeded. But along with that, we had a very, very small, very weak cross-reactivity with an HIV protein called GP41. And that cross-reactivity was being picked up in some HIV diagnostic assays. Now, we anticipated that this might be an issue. In fact, in the consent forms that our participants all signed, we did highlight the fact that this might be a possibility because the clamp, which makes up the underlying core of our platform technology is made from one of these HIV proteins. It's a small structural element, very, very small piece of this one protein, a small structural element that holds the vaccine in place to present it to the immune system in exactly the way it should be presented. And so this very small piece of this HIV protein was eliciting in itself a small amount of antibody reactivity. It was really weak, but it was still being picked up by HIV diagnostic assays. And so we reached the point where we knew this some weeks ago, and we believed that there was a pathway to traverse that particular issue, to navigate our way through the HIV diagnostic pathway. But last week, the decision was made that the communication strategy in particular around that to the community in general was maybe a step too far. And that's also at a time when we'd also heard that other vaccines had shown very strong efficacy in their phase 3 trials. So I think the decision was made that whilst the vaccine looked promising, this particular issue meant that stopping the trial was probably a more effective way, or at least stopping going forward was a more effective way of going forward for the Australian population. We had other candidates that are going to be possible to be deployed. So that's the main reason. We had a vaccine that was highly effective in inducing the immune response we wanted, but the sideline issue of this diagnostic interference was something that was felt maybe challenging to address.
I can't imagine what that roller coaster ride must have felt like, and still probably does feel like. But Trent, I think maybe building on what Paul's saying, this next question is coming from Georgina Hayden. She's asking, why did you choose the HIV virus protein in your molecular clamp?
Yeah, it's a really good question, and there's two main reasons for that. One is the team had done a lot of work on this particular version of the clamp, and all of that work really pointed to some really promising data for how well it worked to do this stabilization, which we think is really important part of that eliciting the right type of immune response that Paul was talking about. So, to get that activity against the virus, we really knew we needed to have a stable vaccine. So that's, and this particular construct worked well. It was also at a time when we were partway through a research program with our partners at the Coalition for Epidemic Preparedness Innovation, and that was the platform that we had proposed as part of our rapid response pipeline. And so that was what we had in the toolkit back in January, and that's what we decided to go with. And I think the final piece of that is we also had another element to this particular clamp that let us do part of the scale-up work. And we knew back in January that the only way forward for any vaccine candidate was something that could be produced at mass scale. And so putting all those elements together just meant that it was the right decision at the time. We hadn't had the opportunity to look in clinical trials at this particular issue before. And it was something, as Paul said, that came up a number of weeks ago. We looked to see what was possible in terms of navigation, but as you heard, those elements of thinking about impact on communication and also potentially on vaccine hesitancy were absolutely critical in the decision. So that's where we landed.
That's really helpful. Thank you, Trent. And at least for me, that explains quite a bit. So we have another question that's from Julianne Wolf, who asks, how does the UQ technology differ from the Pfizer-BioNTech technology? Paul, could you maybe help us with that one?
Sure. There are a number of different flavours to viral vaccines that one can make. Protein subunit vaccines such as ours, so we make a protein, recombinantly express this protein, purify it, and that's what's given to individuals as the vaccine. It's not too dissimilar from the influenza vaccine. Made slightly differently. In the influenza vaccine, they grow influenza in eggs and then break it apart and then deliver those subunit components. So that's one way of delivering a vaccine. Another way is to attenuate the virus so that it doesn't have the same disease-causing effects but is able to stimulate the immune response, and measles and polio vaccines are like that. There are some new vaccine platforms that have been around for a couple of decades now, at least in the research labs, and they've come to the fore in this year in testing against COVID. One is to take the gene that encodes the vaccine immunogen, the bit of the protein that is going to induce the appropriate immune response, and embed that in another virus, but one that doesn't cause disease, and in fact, in some cases doesn't even grow, but it's able to deliver that gene. And that's what the University of Oxford or AstraZeneca vaccine is. It's a chimp adenovirus that's defective, in other words, it won't grow, but it delivers the gene. So again, that gene gets into host cells, the human host cells, and is expressed and stimulates the immune response. The BioNTech vaccine, or the Pfizer BioNTech vaccine, is a next level. It's a newer generation vaccines that base solely on the genetic information itself. And so it's a messenger RNA molecule, so a nucleic acid molecule that's formulated into lipids and other ways of ensuring that it gets into cells in the body, but it's delivered essentially as an RNA molecule. And off that is expressed the protein that is the immunogen. What unites all of them is the fact that they're all targeting the spike protein. So we make it externally and then deliver it. The Pfizer vaccine has the gene encoding that protein in a piece of messenger RNA, which is then delivered.
That's really great, and highly complicated, but somehow you're able to translate that into what essentially feels like layman's terms. So I really appreciate that, Paul. Thank you. Now, Trent, Helen Rennick has another question for us, which is, it sounds like we've learned quite a bit about some incredible advances here, but what are the other uses for this research, or this COVID research?
Yeah, so I think many areas of science have been just propelled so quickly this year, and the same is true for what we've learned for the molecular clamp platform. I mean, the opportunity to move very quickly, work with commercial partners, work with networks all around the world to see how fast we could move. I think we've learned a lot of lessons there. And we've also shown proof of principle in people in this clinical trial to validate essentially this idea of method of stabilizing this vaccine with the clamp. So that's absolutely critical. I think important for us is that there's a number of opportunities to make next generation versions of this technology that take away those regions of the GP41 from the HIV virus that was used and replace them with other elements from different viruses of which there won't be any diagnostic interference. So I think that's a very promising way for us to move forward, and we're very excited to continue that work. So that'll form the foundation of quite a bit of research at University of Queensland going forward. So we're very excited about that. And then I think on the Australian level, we've really shown what happens when people get together and do amazing science. So we're also very excited about a potential to create a vaccine research alliance across the country with a large number of leading infectious disease researchers and groups. And so I think, hopefully you'll be hearing a lot more about that. It's something that we're quite excited about. And I think it's a real opportunity for Australian research to find a way of getting clinically validated, move forward, and hopefully create solutions, for the next challenges that come forward, whether that, not that we want to think about it, whether that was another pandemic or whether it's things like multi-drug resistant bacteria. All of these challenges exist, and infectious disease researchers spend their time and energy really trying to solve these problems. So we're hoping that's something really positive that comes out of this.
And now, Paul, I imagine you might have something that you might like to add to that as well. I mean, you both have been incredible partners on this journey. Is there something you'd like to share?
Yeah, perhaps just building on what Trent's just said, we're essentially already starting to work on Clamp 2.0. So the next version, we're taking the strong positives that I mentioned before that came out of the phase 1 clinical trial to underpin the strength of the platform technology. So we're confident in going forward just with that change in from a GP41 backbone to a different backbone that won't have that or engender that sort of interference problem. But I think probably the biggest outcome from this year has been the realisation that Australia had some sovereign gaps in its capacity to address the outbreak as it happened. I mean, we've done extraordinarily well in terms of public health measures, but in terms of the underlying research and development, while we have an extraordinary amount of expertise in this country in virology and immunology and vaccine design, we have some gaps in moving that towards a commercial arena to translate it to actual products, to vaccines. And with that realisation and the work that we've been able to do and others in this space in a very collaborative way across state borders, we've had a very strong connection with the University of Melbourne and the Australian National University in this project. We believe that there is a way forward in generating a vaccine alliance that will hopefully progress this and other vaccines a little bit further down the path than previously has been possible. So I think there's a lot of learnings to come out of this, and we have really positive directions to move forward.
That definitely sounds very promising. Thank you for that. And actually, maybe building on this is another question that's coming from Kwan Hung Ong, which is, what is the next step for the research group after the withdrawal of the vaccine project? Who would like to take that one?
Yeah, well, I guess we've just talked about some of the next steps we're taking with the research itself, but we shouldn't forget that we have a phase 1 clinical trial still underway. We will be monitoring those participants all the way through into Q3 next year, looking at responses. We may find in the not too distant future that weak reactivity against that is causing the diagnostic interference might actually disappear. So all of these things will inform our next stage of the process. But I guess we'll be focused not only on that but also on progressing the underlying technology and the partnerships we've built.
Yeah, and I think it's important to say too, we've got a lot of research that was done in a very intense period, and we're really looking forward to, as scientists, putting that out and publish that work and get it out into the scientific literature. So that's something that's also very important. We think we have a number of learnings in our data that are important for everyone to see, and we're looking forward to sharing that in some real detail. We've been, as Paul mentioned, working with our partners, looking at some of the deeper immunology around what this vaccine looks like. And of course, we used a technology from CSL Seqirus, an adjuvant technology called MF59, and we're hoping that there's some really important insights that come out of the work that we've done. So that'll also be a big part of the work. So it's definitely, nothing's really stopping for us. In fact, it's probably just a continuation. We're all looking forward to perhaps taking a little bit of time off over the Christmas period, trying to rest and recharge, but that work will be critically important, and we're working as fast as we can to get that out.
I suspect everybody who's tuning in right now would be encouraging you guys to really enjoy that time off after the last few months. So it sounds like this isn't the last that we'll be hearing from you all. In fact, I think you might just have a bigger community of supporters looking to see what you publish. So that might lead me to our final question, which is from Peter Ellerton. Peter asked, what's the most important learning from the team? I'm sure you both would have some valuable insights to share. Can I ask you to respond?
Yeah, I think we probably addressed some of this before, but for me, the biggest learning out of this year has been the power of partnership. And, that we began that journey a number of years ago in reaching out and collaborating with our colleagues on our CEPI-funded project in Melbourne and in Canberra, but also, our CSL friends and all of the contract industries that we've been working with as well. That partnership has allowed us to progress at the speed with which we have moved, along with the generous funding support from our donors. All of those things have combined to allow us to progress to where we are. So that strong partnership, I think, is a foundation for what we can see into the future. As I mentioned before, we're looking to build a vaccine research alliance, which will hopefully build on that and hopefully engender maybe even a new industry within Australia in this space.
Yeah, I completely agree, and I think the other part of this year has just been watching in awe, really, both with what our team has done and what hundreds and maybe thousands of teams around the world have done in the scientific community, that is, in what we've been able to achieve. I mean, we went from a position back in January where it really wasn't known whether it was even possible to develop an effective vaccine for humans for coronavirus. And now, with more than 300 vaccines in development and several vaccines that have shown incredibly promising efficacy data in massive clinical trials, that's incredible. And then the underlying understandings of what has come from just studying this particular virus, I think we'll have learnings for decades to come. I think the number of PhD projects that have been essentially covered in less than a year this year is going to be astounding. So it's been an incredible year for science. And then on that, I think the other piece has been watching how the public is engaged with science. I think science communication is very complex. Sometimes science can seem just foreign and far away, but I think there's really tangible, important things coming out of what science has been able to achieve this year. And that's why I think communication for us this year has been critical at every step. We've really wanted to talk about the work that's going on here, and I've seen that from all the groups around who are doing this type of research and others. And so it's been fantastic from that perspective when we're looking forward to science continuing to provide solutions for some of the most challenging things that we face.
Yeah, maybe if I can just add to that too. I'm just thinking about other learnings out of this experience, and I think a very important one is the experience we've just been through, and that is that the process itself is open and transparent, and I think the community can believe in the integrity of the processes we need to put these vaccines through. We got to a point where there was a realisation that perhaps going forward was not the best option, and those decisions are always made when the process has integrity embedded within it. So I think confidence in the approaches. There's been a lot of commentary around the concerns about safety given the speed with which these vaccines have progressed. And the message I've been trying to give all the way along is that no corners are being cut in terms of safety. We've cut a lot of other corners in terms of manufacture and so on, but not in terms of safety. And I think our experience is probably a testament to the fact that the appropriate checks and balances are in place.
Well, I know, 2020 hasn't kind of panned out the way any of us expected, and certainly your team and anyone who's tuning in today. I will say on the advancement team's end, working with the two of you and the team behind you has been really extraordinary. The way that the community has rallied behind the science that's underway, their genuine interest in trying to understand and to be advocates as well for this team has been extraordinary in each and every way. It's unlike anything that I've ever seen before. And while we've never really met with many of these people in person, including many of the people who are on this call, we have had quite a few of these chats together online or on Zooms, multiple Zoom calls. And I just really want to thank both of you for investing your time and really making the most out of this opportunity to also bring community scientists and community supporters on this journey. I hope you felt that support throughout. Now, with that final question, it's time to unfortunately draw the question portion of this discussion to a close. Although I might, just since I have the liberty to ask you one more question personally. As I mentioned, this project has inspired truly a global community to stand behind you. Many of you who are on this call, and of course, Paul and Trent, are very well aware of the beautiful letters that have come in, the scientists with superhero capes drawings that are on your lab's walls, as well as, from these coin jar donations to the multi-million-dollar donations, which are just extraordinary. But all in all, over 2,600 people, foundations, corporations, and of course the federal and state government came together to showcase mateship in action. So personally, as I've shared, it's been a really humbling experience, and I hope the same for you. But what would you want to say to those supporters, since many of them are already on this call?
I mean, a huge thank you. I think I opened up with that thank you. I do it again, not only to our supporters who funded much of the research program, so from the donations from foundations, but also down to the coin jar donations from children in Melbourne that we received, and it's also just the community support we received. So through responses to media articles or cards that we've received. I mean, I think one of the most surprising things for us since Friday, when this was announced, I was dreading the announcement because I wasn't sure how people would respond. They'd put so much faith in us, and almost without exception, what we received is just an overwhelming volume of email and cards, and all expressing their support for what we've done. Some wishing that we were still progressing with that work. We've also had remarkably balanced media coverage, and so we thank everybody for all of that support. But before I progress any further, I just want to shout out, I mean, Trent and I are here today, but the broader leadership team, Keith Chappell, Dan Waterson and Christina Henderson, and the two of us have really been pouring a lot of time into this particular program. Keith in particular has committed so much of his time, and I imagine all of us have families that have been impacted by the commitment as well, but also it's the broader team. At UQ and our collaborators. So I just want to shout out to them, and also a special shout out to all those people who put their hands up when we called for participants in the vaccine trial. I know when there was an amazing overrun of potential participants, many people who didn't get the opportunity to enter into the first part of the vaccine program, but the second part, when we called for those above 55 years of age, within 24 hours we'd received 7,000 requests to be part of the program. So it just showed that level of community support. So thank you all to all those who put their hands up, and all those who actually did end up entering into the vaccine program.
Yeah. So I would just reiterate exactly those sentiments. I think you said it really well, Paul. I think it's probably fair to say that the support from the community, whether it was financial support or those letters of support or emails, helped the team during some of the toughest times where, we were facing big challenges, technical challenges at the start of the year, financial challenges in the middle of the year, timeline challenges all the way through. And I think those messages really helped drive us forward. And I think we feel very proud to have hit every milestone we set ourselves this year, which I think is quite amazing. And secondarily, I think, to the team, absolutely heroic effort by so many people here at UQ. I mean, just because we were with those people all day, every day, whether it was during lockdown when there was no one else on the roads but us going into work, that was a really strange period. But to our colleagues and collaborators around, both around the country and around the world who did heroic things this year, small companies, large companies, service providers, you name it, those people just really, when we asked them to do something, it wasn't, oh, that was too hard. It was like, okay, we got to find a way to get this done. So all of those things, absolutely incredible. So just with that, just a lot of thanks, really. Definitely, not exactly where we wanted to be, even though we feel quite proud about the achievements. But I think, we can all be, incredibly proud of what you guys made possible. So thank you.
Very, very well said. And I might say two things are certain. We have a really, truly extraordinary team and an extraordinary university right here in our own backyard that's doing incredible science. And we've gotten to see that up close and personal this year. And so thank you to all of you on the team who have contributed to this and who have helped put UQ and the work that's being done in the spotlight. And then also, I think the other big piece is, just as you said, Trent, without the support and generosity of the UQ community, whether near or far, I don't think this project would have been quite as possible as it ended up being. And the learnings and the learnings that surely are coming out of this will be ready and available for the future. And so I just think it's amazing what you've done. And I'd like to personally express my gratitude to both of you, to the team behind you, and to all of you who partnered with UQ on this journey. If additional developments occur, you will be surely amongst the first to know. Paul and Trent, thank you to you and the whole vaccine team again for the sleepless nights and sacrifice and driving in when all of us were tucked away at home. Your commitment is truly extraordinary, and I do hope the holidays bring some well-deserved rest. We're incredibly proud of the work that you've done, and we know that UQ community will continue to be celebrating your efforts and acknowledging the historic effort that was put into this. We look forward to seeing the continued impact. Now, on behalf of a whole community of supporters, we have a little bit of a surprise for you, and that includes 2 special messages from supporters we want to share with you today.
My name is Georgia Smith, and on behalf of all the girls at St Margaret's, I just wanted to say how incredibly proud of you we all are. We know that you have put so much work and effort into this vaccine, and we cannot say thank you enough. Through our school's interest with this vaccine, it has taught us a lot more about science and the trial and error nature of science. Thank you so much. You should be extremely proud of yourselves.
Hi, I'm Jenny from the Paul Ramsay Foundation, and we wanted to extend our thank you to the UQ team working on the vaccine. You've been an inspiration to all of us. We are very proud partners. We have been and will continue to support you and be behind you all the way. The landmark discovery that you've made using the molecular clamp has been a milestone in scientific discovery, and we look forward to the long-term benefits that this will bring to the vaccine development space. Thank you very much, and we will continue to support you.
Thanks to Georgia and Jenny for that. And we have been receiving quite a few comments throughout this discussion. I just wanted to pass on one from Amy McCart-Reed, which is, thanks so much for your efforts. Super reassured that our rigorous standards are applied, but also disappointed for you guys. It's been a great source of pride for me to tell everyone that Paul was my undergrad lecturer and that Trent and I shared a PhD student office in Building 76. Here's another comment from Alice Evans. Congratulations on the journey undertaken, the relationships developed, and the research achievements. While there is a sense of disappointment, you are all to be applauded. Another one from Jeff Martin. He says, congratulations for being open, honest, hardworking. You have maintained your integrity, and my admiration for the work you are doing has just grown. Now, there's a whole pile of additional comments that we've been receiving and collating and kind of drip feeding to the 2 of you to share with your team. And I know you've been getting a lot from the community as well and doing your best to respond. But you know, it's been, as you've shared, just absolutely inspiring to see this group that is standing right beside you through this roller coaster ride. Do you have any other final comments or words you'd like to share with the people who are joining us today?
Thanks to everyone. I need some sleep.
Yeah, and same for me. Look, really, it's just thank you.
Yeah.
You know, and it wouldn't be possible without your support and words of encouragement. So really appreciate it.
Absolutely.
Yeah, fantastic.
Thank you all.
Well, while this year's been fraught with challenges, watching the community come together in support of this team's research has been truly incredible. So to those of you who've joined us today from all around the world, please have a good afternoon.